All right, we want to get started here. Happy Thursday, and want to welcome everyone formally to Season 3, Episode 1 of CRISPR Office Hours. Thanks for joining us today. As we've been hosting CRISPR Office Hours for about 2 months or so, we've been doing this to help the genome community have a venue to connect, ask questions, and make sure we navigate this pandemic together and tackle the challenges that are before us as a community. And as a result of it, we've had great speakers, we've had great panelists on here and covered a lot of topics that are relevant to the community and addressed a lot of the emotional issues that come from conducting shelter in place and that the pandemic has resulted in. So for housekeeping purposes, this CRISPR Office Hours will be recorded and we will be having it on our YouTube channel. And there'll be a blog up afterwards also with highlights of the actual office hours and a link to the YouTube video. And you can go to synthego.com/blog to look at our previous office hours also as well. And the key part of this is to ensure that it's very interactive. So please ask questions in the chat. Please make sure to mute yourselves and ask questions so we can address them. And that way everyone has equal opportunity to ask and get heard. So with that, let's get started here. My name is Aditya Vempati. I'm the VP of Marketing at Synthego. As always, my amazing co-host, Kevin Holden, along with our guest panelists, which we'll introduce.
Hi, Aditya. Hi, everybody, and welcome to CRISPR Office Hours. So I'm your co-host, Kevin Holden, Head of Science at Synthego. The title for our episode this week is Accelerating an International Effort Led by UCSF to Identify Drug Targets for COVID-19. And I'm also joined this week by Jared Carlson-Stevermer, lead research scientist at Synthego, who some of our longtime viewers may remember from his last appearance on Season 1, Episode 2. So, hey, Jared.
Hey, guys. Great to be back. Feel a little bit like a guest host on SNL, where I got to keep racking up the appearances and get into that secret green room.
All right. So, our mantra over the past few months here has been to keep calm and carry on. And our version of it here at Synthego is to keep calm and CRISPR on. So please stick around until the end of today's episode, where we'll let you know how you can get a special edition Keep Calm and CRISPR On t-shirt.
So without further ado, I want to introduce today's panel. Kevin, Jared, and myself are excited to welcome Professor and Director Nevan Krogan of the University of California, San Francisco, and Chief Operating Officer of UCSF's Quantitative Bioscience Institute, Jacqueline Fabius. Nevan and Jacqueline, it's great to have you here.
Thank you for having us.
Great to be here.
So as we do with all office hours, we want to make sure, again, it's as interactive as possible. We get the audience engaged and make it fun. You know, we want to make sure people feel welcome to ask questions, engage with us. And one of the things we like to do is introduce a poll question. So the poll question for this week is, what's your go-to emoji during the pandemic? Is it number 1, dancing emoji? Number 2, shrug? Number 3, CRISPR, DNA and scissors. Number 4, coronavirus, that's what we're depicting it as. Number 5, toilet paper. 6, scientists. 7, face mask. And then 8, alcohol, wine, beer, whatever you prefer. So I want to ask Jacqueline and Nevan, what are you guys mainly using when you communicate?
I think I'm always holding the glass of wine. I don't know if I'm always using it, but going for number 8.
This one's easy for me. It's definitely number 1. Because I'm a hell of a dancer.
What about yourself, Kevin?
Yeah, I think during this time, it's like, and everything else that's been happening in 2020 is probably number 2, the shrug.
The shrug. What about yourself, Jared?
Yeah, I mean, I just got both my answers taken. I was going to go with the shrug and be like, yeah, what's happening? And then also respecting social distancing with a good, cheers from afar.
Yeah, I think I started shelter in place with a shrug, and I think I am now definitely in the wine and beer emoji reaction to a lot of things. So just seeing a lot of our audiences, we're hearing a lot of folks hit 7. face mask emoji. So that's good. I'm glad to hear people are either telling others to wear a face mask or they're saying they're wearing a face mask. Either way, I think we're in good company here.
Do we only have one answer?
No, we have a bunch of answers, but majority are saying face mask. There's a few that are saying shrug, and then a few that are saying beer, wine, and alcohol as well in the chat.
Yeah, it looks
So-
looks heavily skewing towards 7 and 8.
Yeah, definitely 7-8. Face mask and drinking.
We got Juan Pablo with a number 1. He's dancing with me.
That'd be a fun time. So with that, I want to move on to getting started here. So Kevin,
Okay.
you want to kick us off?
Sure. Yeah. Okay. Let's get started with the discussion today. So, Nevan, actually, and Jacqueline, can you tell us a little bit more about the UCSF Quantitative Biosciences Institute? What's the scientific vision behind it, and what makes it kind of a unique collaborative environment?
Sure, maybe I can start and Jacqueline can add. So the Quantitative Biosciences Institute, as you say, is an institute at the University of California, San Francisco. And I would argue that if there's one word to describe it, it is collaboration. So our vision really is to bring people together, break down barriers, break down silos, and get people working together on important biomedical problems. The focus of, a lot of the focus of the Institute is really on technology and bringing technology together in a disease agnostic way and focusing it on a number of different important disease areas. And we're trying to bring together scientists here at QBI and at UCSF. We're trying to connect different scientists here at UCSF across different disciplines. And we're also trying to make connections between scientists around the world. This is a lot of work that Jacqueline and I have done over the last 3 or 4 years. And I would say because of that, we were in a very good position to respond to this pandemic that we're in right now. And this is what we're going to be focusing our talk on today. I don't know, Jacqueline, did you have anything to add to that?
Maybe I'll let you go into one little bit more in the science and then I'll add to that.
Sure. Okay, we'll get right into this here. So as I was alluding to, we're really technologically based and we're bringing together diverse technologies, I think in a very exciting and integrative way. On the left here, including mass spectrometry, looking at proteins, genetics, CRISPR-based genetics, structural analysis, now using a lot of cryo-EM, and then combining this also with chemical biology and chemoinformatic approaches. And I would argue that we try to put all these different technologies together and get at the intersection of the data sets. If multiple data sets are telling you the same thing, this is where you should put your focus. And this is kind of the strategy that we've employed. And as I said, these are obviously disease agnostic approaches. And we've been focusing them on a number of diseases in the context of these cell map initiatives, which are on the right, the cancer cell map initiative, the psychiatric cell map initiative, and then the one initiative that's obviously pertinent to this particular presentation is the host pathogen mapping initiative. And the idea here is we're using these approaches in an integrative and unbiased way to study how different pathogens come in and hijack and rewire the host during the course of infection with the ultimate goal of coming up with novel therapy. So maybe before I go to the next slide to get into some data, Jacqueline, did you want to add to the vision of QBI, the way you see it?
Well, I think that QBI is really entrenched in both curiosity and creativity, and I think that's how we approach everyone and everything we do. There's the creativity in science, and QBI especially has also used this creativity to broaden or get the message of science out to both other scientists in the community and to lay audiences. And that's a real sort of marriage of creativity that comes together there with the scientists. The curiosity, I think, leads not only the science itself, of course, that's, I think, the sort of cornerstone of science, but also how we lead our research. And I think Nevan's going to go into that and how we develop the maps.
Great. So if we go to the next slide. So one of the tools that we're heavily involved in, as you saw on the previous slide, is mass spectrometry. And we use mass spectrometry to identify oftentimes protein-protein interactions. And we've been doing this on a number of different viruses over the last 10, 12 years. This is where we simply just take individual viral proteins, put affinity tags on them, express them in cells, purify them, individually and then see what human proteins are physically associated with each of the pathogenic proteins. So here are a number of maps we've generated over the last several years, including in the top left HIV. If you go to the top right there, a number of different enteroviruses. In the middle there on the left, Ebola. We have a very large program, influenza, which is to the right of Ebola. And then in the bottom right here, we're also looking at a number of flaviviruses, dengue and Zika. And then there's a couple of bacterial species here. We often analyze the secretome of the different bacterial the different bacterial species that are often problematic for mankind. And normally these type of maps take a long time. I say 2 to 3 years per map. It's at least a year and a half. But we generated with these tools a SARS-CoV-2 human protein-protein interaction map in a matter of weeks. And to me, this is a testament to the collaborative spirit that went into studying COVID-19. And that's one of the remarkable things about this pandemic, I would say, is how scientists have come together in an unprecedented way. And I would argue that QBI was in a perfect position for many of the reasons Jacqueline outlined to respond to this particular pandemic. Next slide.
So I could give you a bit of a background on QBI, and as Nevan said, why we were so well situated to allow for the formation of a group like the QCRG so quickly, the Quantitative Biosciences Coronavirus Research Group, QCRG. QBI is, there are 103 labs affiliated to QBI focused on collaborative research. The research is quantitative, it's discovery science, it's disease agnostic, and experimental and computational. As Nevan mentioned, we're known for our cell mapping initiatives, and these are initiatives that have 10 to 15 PIs each, so 10 to 15 scientists or investigators working in their labs. So essentially, when you look at these 3 initiatives right there, you're talking about hundreds of scientists working together on specific problems and each bringing their own expertise to the table. We are, however, also mostly known for focusing on young scientists and empowering women. And I think what really makes our reputation at home and around the world are our very unusual and successful events, all kinds of seminars, and importantly, international symposia, as well as regular domestic symposia that bring together scientists from everywhere to exchange ideas and to start collaborating. Next slide. So our culture of collaboration really started at home with these cell mapping initiatives. And we had such a success that we started thinking, well, what if? What if in the same way that we map the cells, we started mapping the world with our collaborative interactions? What if cell mapping, the maps that you saw earlier, for example, equaled world mapping? And what if protein-protein interactions also equaled people-people interactions? And as such, next slide, Nevan and I started looking at the world and where we had been and what that meant. Next click. And so we looked at our activity around the world and the relationships we were building and further thought about, well, how exactly are these networks and how are they forming? Next slide. And so in a very similar way, we saw that we started having these people-people interactions. And you could group them around specific interests, whether that's infectious disease, technology, cancer, cardiovascular, and so on. And importantly, for the story that Nevan's going to tell you about under infectious disease, you'll notice that both Paris and New York were already in our network. And as such, we built these incredible relationships and built further, next slide please, and started establishing formal relationships with some of the best places in the world to eventually bring the scientists together. So we have connections and collaborations in Germany, Ireland, France, Nigeria, Poland, Israel, the UK, and importantly, the Institut Pasteur in France, which Nevan will be mentioning a bit later. And so all of this really leads to the next obvious step, which is the formation of the QCRG.
Yeah, Jacqueline, if you don't mind, could you tell us a little bit about how you leveraged all these different connections and really made the coronavirus research group?
So the formation of the QCRG was really understanding the context of a coming pandemic around February and the scientists pivoting immediately to address it. So we had we had meetings late at night in March where scientists came together to decide to move their labs immediately to focus on COVID-19. And the discussion was, okay, who do we have in place? What can be done? Who can do what? And so we knew we didn't have the virus at UCSF or in place at the time. And we immediately looked to our network of collaborators. And so what stood out was Mount Sinai in New York and the Institut Pasteur in France, who had the live virus. And because of these deep relationships that we had already established, it was essentially just a phone call, right? And because you have this culture of curiosity and collaboration, people were immediately on board. I'm not sure Nevan finished his sentences before people were on board. And so next slide. So within really a couple of days, you had an initial 22 scientists who gathered at UCSF to focus their research on COVID-19, and Nevan will, I think, tell you a lot more about their science.
So it initially started with 22 labs, and then as you can see, it expanded to over 40 labs, essentially encompassing hundreds of different scientists coming together, I would argue, in a way I've never seen before, in a very unprecedented way, bringing together their integrative tools in a very exciting way. So we had systems biologists here, structural biologists, chemical biologists. Can you go back one slide? Computational scientists and virologists all coming together, bringing their tools to the table to try to come up with solutions to fight this particular pandemic. So the group got so large, next slide, we actually had to break up the QCRG into different subgroups, and these are the leaders of the different subgroups. We were on these Zoom calls with hundreds of scientists. Now we've broken this up so that the meetings can be more effective. And you can see that the different subgroups focus on different technological areas like structural biology or bioinformatics, or specific biological areas like translation. There's David Ruggiero, ubiquitination, John Gross, and protein trafficking, Cherie Makarigi. So these subgroups have formed, and now what we're doing is having a lot of crosstalk between the different subgroups as we progress our research on COVID-19.
Yeah, so actually, Nevan, can you tell us about now the SARS-CoV-2 interactome study and what inspired you to start the study?
Sure. So we had been looking at viral host interaction maps, as I alluded to before, for many, many years. So we were in a very good position to generate a SARS-CoV-human protein-protein interaction data set. And in January, David Gordon, a very talented research associate, along with the technician, Gwendolyn Zhang, in January, they started cloning out each one of the genes. This was using a sequence from, I think, the first person that was confirmed to be infected in the United States in the state of Washington. They started to clone out these genes, and when it was realized that this was going to be a huge problem around the world, more and more scientists got involved. And we generated this map, as I said, in a matter of weeks instead of a matter of years, as is what's normally done. And showing you here on this next slide, the vision here, sorry, go back one slide. The vision here was to try to understand with this map how the virus comes in and hijacks and rewires the host during the course of infection, trying to focus on the host instead of the virus. So there's a lot of therapeutic studies ongoing, trying to find drugs or compounds that target the virus. We're taking a different approach. We're trying to look at the host, try to come up with drugs or compounds that'll target host proteins the virus needs in a drug repurposing efforts. And there's advantages of doing this. You don't have to worry so much about resistance. We don't mutate as fast as viruses. Also, we and others have shown that viruses, and very different viruses, sometimes target similar human proteins. So if we come up with a strategy for COVID-19, the idea would be effective for COVID-22, COVID-24, and other viral infections that come online in the future. There are disadvantages here on the right, and one of them is toxicity. But however, we're focused initially on drugs and compounds that were either FDA approved or in clinical trials that have passed toxicity issues. So hopefully we're looking at compounds and drugs where toxicity wouldn't be such an issue. So on the bottom there's about 30 genes in the virus. As I said, we cloned out each one of these one at a time, expressed them in human cells. And our cells have over 20,000 genes, and the virus can't exist by itself. It obviously needs our genes, our proteins, our cells in order to live and replicate and infect us. And through our protein-protein interaction study, we identified over 300 proteins. And then we narrowed down, and great collaborations with Kevin Shoket and Brian Shoket here, fantastic chemical biologists. They identified 69 different drugs or compounds that would target at least one of these human proteins that we think the virus needs. And then we're trying to figure out if any of these would have antiviral effects. So next slide.
So Nevan, just real quick, if people are interested in studying SARS-CoV-2, are these available?
Yeah, we have. So we initially put out our map. If you can go to the next slide, I can just allude to that. We generated this map, we put it out on bioRxiv at the end of March, and then we tweeted out that we had all of these genes synthesized for the entire virus. We said we'll give these out for whoever wants them, and we sent them out to, I think, over now 350 labs in 40 different countries, 180 different cities. So we've been sending these out free of charge, no strings attached, no MTAs, and we're still getting emails. So if anybody wants these plasmids, just please email me, and would be happy to send those out. And we're very happy that we were able to, around the world, expedite research on SARS-CoV-2. So hopefully others can come up with strategies to fight off this infection. So here's the map that we initially had put out. And again, it's 332 SARS-CoV human protein-protein interactions, including 69 different druggable host factors. The red diamonds are the coronaviral proteins, and then the nodes, the circles, are the human proteins. And if we think they're druggable, they're in orange. And I just want to point out here on the bottom right, we collaborated with a company in Hollywood called Zoic Labs. They're actually involved in making movies. And Ron Conway and Biz Stone, the founder of Twitter, who have been helping us philanthropically, got us connected with this particular company. And they've made this map very interactive using really cool tools. So if you're interested in this data, I'd encourage you to look at this website and look through the data in a very interactive and intuitive way. And we're continuing to work with them, putting more information in their tool so that people can peruse it much more effectively. There's actually a piece in the LA Times that just came out yesterday describing how Hollywood is working with biomedical researchers on COVID-19. So I think that's pretty cool. Next slide. So the idea here then was to take this map, and we worked, there's the pictures at the bottom of Kayvon Shokat and Brian Shokat. They identified these 69 different drugs and compounds. It's a mixture of, I think, 29 of them are FDA approved drugs, a dozen were in clinical trials, and the rest were being studied preclinically. We wanted to test them to see if they have any antiviral effects. As Jacqueline said, we didn't have the virus propagating or growing here in the Bay Area. Melanie Ott is working very hard to get the virus up and running here so we can do these screens. We're starting to work with her. But we started to leverage our collaborations that Jacqueline and I had set up over the last 3-4 years, including with Mount Sinai. And there on the left and the bottom is Marco Vignozzi, a picture of him. And then on the right, Adolfo Garcia Sastra. And we sent the drugs and compounds out to them, and they have been testing them. And the original paper we published, we tested about 2/3 of these, almost 50. And we're focusing our attention on a number of drugs that we're very excited about that have antiviral effects, and we're going deeper into the biology of them.
So,
And I'm sorry.
Oh, I was going to say, just to follow up on that, did you uncover any of these targets out of the interactome that really stuck out, or something that you were very interested in following up with?
So the couple of areas we're focusing on right now. One, are proteins involved in translation. So the virus needs our translational machinery, obviously, to make its own proteins. So we're focusing on a couple of drugs and compounds in that arena. And then because there was a number of interactions in the map that pointed us to translation. Then there was another, and Kayvon Shokat's been leading the charge on that. Another connection on the map is with these receptors called sigma R1 and sigma R2. And Brian Shokat here at UCSF has been working on those for a long period of time. And there's a number of drugs and compounds that are known to target those receptors that have, really, many of them have potent antiviral effects. And we're really focusing our attention there. Genetically, we've shown now that these receptors are important for infection. And what's so great about these receptors is that they're very druggable. And there's a whole slew of FDA-approved drugs that are known to target these. And now we're just marching through those and trying to find the best ones that we could ultimately take into clinical trials. I think Jacqueline was going to add something too.
Well, I was just going to add a little bit of a backstory to this, which is, how far collaborations go and what that means. And so when we were about to ship the drugs and compounds to New York and France, obviously it was not a problem with New York, but we were looking at flights and things stopping to France. So there was an issue getting the drugs and compounds to the Institut Pasteur in Paris. And we have a good relationship with the French consulate here due to events we did with them. And they called immediately to, to ask what they could do or anything. And in fact, the French consul here called the French ambassador in DC, who called the head of FedEx and also called the French customs top people to make sure that our packages were not stuck in customs and would get to Pasteur really easily. So that's just a little bit of the human backstory in this.
Power of networking. Nevan, just real quick, we had a question come in from the audience here from Matthew, who was asking, would these compounds simply prevent the virus from binding to these targets, or do you have any idea how that would work?
The map that we generated is intracellular, so we don't think it's the connections we're uncovering are connected to entry. We think it's post-entry, and all our virological data definitely points in that direction. The receptor I alluded to, sigma-1 and R2, they're actually on the cell surface, but they're also intracellular. And we think that the regulation is happening. There's certain viral proteins that are hijacking these receptors. When they're in the cell, they're actually on the endoplasmic reticulum, and we think that's how they're ultimately mediating their effects with respect to infection. So we're looking closely at this area, as I said, because it's very druggable, and we're very hopeful that we'll come up with something that'll be useful in the near future.
And we had a quick follow-up question from Raul in the audience, who asked, of those 69 FDA-approved compounds that are already commercialized, are any of them, I guess, what was the strategy to choose those drugs? That was the question. And are these, what is the strategy to choose those drugs?
Well, we looked at the proteins on the map, and then we said, okay, which drugs or compounds are known to bind to those proteins or inhibit those proteins, obviously for other diseases, mostly anti-cancer drugs and compounds. So the logic was, all right, let's take some of these drugs and compounds that are obviously been developed for other diseases, but since the virus uses those same proteins that those drugs or compounds are known to target, maybe they'd have antiviral effects. So there's a lot of people doing these kind of large drug screens, tens of thousands of compounds, which I think is great. We've taken a different tact. We're like, okay, let's understand the biology of infection, and let's let the biology point us in different directions with respect to the pharmacology, the chemistry, and ultimately therapeutics. So instead of screening tens of thousands, we screened less than 100. We've got a couple categories we're excited about, then we can augment that chemical space. And because you know the biology, you can go back and forth between the chemistry and the biology so much more effectively. And I would argue that puts you 50 steps ahead with respect to developing novel therapies.
And then we had a follow-up question too from Catherine, and also Mina asked a similar question about the types of cells. Are you using cells that express the receptors?
Yeah, initially, it wasn't clear which human cells would work best. There's still a lot of ongoing research around the world to determine which human cells are and this is related to our collaboration with Synthego as well. Initially, we did African green monkey cells, Vero6 cells, because those are easily infectable. And we've generated a lot of great data there. More recently now, there's been some progress, you'd say, on human cells, such as CACO2 and CALO3 cells. But our collaborators in Paris and New York have actually got A549 cells expressing ACE2, which is the receptor on the cell surface that the virus needs to infect our cells. And these look very infectable. And this is just maybe kind of a segue to the work we're doing with Synthego. We're now systematically using CRISPR to knock out each one of these genes in these different cell types, and then collaborating with New York and Paris and seeing virologically which ones, when perturbed, have an effect on infection.
Yeah, so just to follow up on that, and obviously this is a great segue for us, but can you talk a little bit about how our collaboration with you guys started, and how you potentially see this as accelerating the next step in those research studies?
Well, if I recall, about 100 years ago,
it certainly feels like that.
It feels like that. There's an email that I think that came from Stephen Floor, who introduced us to one of you. And what I,
Oh, I think it was to Kevin.
Was it Kevin? And what I recall is that pretty quickly we set up a call and had a very enthusiastic call with the two of you about what could be done. And well, this extreme enthusiasm on your part to want to collaborate and take part in the CRISPR side of things. So that's how I remember our. our friendship starting. Is this what you recall as well?
Yeah, it was a great conversation, and I remember seeing these two guys, Jared and Kevin, thinking these guys are kind of young. Are these guys legit people to be working with? But it's been great working with you guys. As Jeffrey says, you've been incredibly enthusiastic, and we've made a lot of great progress. And we're starting to get data now, and I'm super excited to see, genetically, which ones of these genes are most important. And you're alluding now to these papers. So on this slide, so on the left-hand side, this is a paper that was published a few weeks ago. That's a study that we just described. And now we're screening through with Synthego, knocking out each one of these genes and looking at effects in infection. There's a paper on the right that'll be coming out soon. This is a complementary study where this was led by Mehdi there, the first author, collaborating with a similar group of scientists, especially Pedro Beltreo in EBI, EMBL in Cambridge, England. We globally looked at phosphorylation by mass spec, trying to then identify or infer the kinases that are being most regulated by SARS-CoV-2. And we have identified a number of kinases, many of which are druggable. I mean, the kinases are probably the most druggable set of proteins in our cells. So there's a lot of anti-cancer drugs out there for that. So we're looking systematically at all these drugs now, and working with Synthego, we're now CRISPRing out in these cells that I alluded to, these different kinases, to see what effects they have genetically. And then the idea is to marry that with the pharmacological data we're collecting as well.
So I think on the next slide, maybe Jacqueline, you can tell us a little bit about, you have quite a lot of other collaborators as well, right, besides Synthego?
Yeah, we do. I think you're on the right path when a lot of people want to come in and collaborate, but not only just to be in and seen, but because they each bring something special to the table. And I think this is what we've been seeing with the various academic institutions, and just people either reading the paper or hearing about the work and calling either Nevan or Kayvon or Brian, usually as an entry, and saying, I want to do this, I can do that, can we get the cells and do, figure this out. So that's been happening on the academic front. On the industry front, just like yourselves, we've been seeing an incredible willingness to come in and help or move things along, right, during this great human catastrophe that we're living, or I don't know if it's a catastrophe or tragedy, but it's certainly something we're all experiencing. And so we've had different things happening with, like, for example, Benchling is offering free lab notebooks to a group that's actually come out of the QCRG. So the QCRG itself is now making its own children. And there's a structural biology consortium that started and already has, I think, well over 60 people that are just specifically in that subgroup. And so Benchling has come along and is offering the free lab notebooks. Effector we're working with, and hopefully going to have something go into clinical trial with them. That's still being worked out. Roche, multiple different projects are starting to percolate with them. And Veer has come through not only with a donation to help the work along, but also we'll be collaborating with them in the lab in different ways. And as Nevan mentioned, Zoic Labs, I think is an excellent example of this creativity and curiosity we were talking about before. These people have never done anything scientific whatsoever. And when we got on that first call, it was, let's see what we can do. And it was a giant, I would say gigantic group of people on the call, like 20 people on the call, and immediately deciding, okay, we could start visualizing this, and that. And I encourage everyone on the call to go on that map, whether you're a scientist or not, because it's so cool. You can just click on the different proteins and see what they're related to, what drugs affect them, what other diseases they relate to. It's a very cool tool that's continually growing on a daily basis.
So yeah, it's fantastic. So Nevan, maybe go into the next slide, back to Jared's question. How do you think the collaboration with Synthego might help to accelerate the next step in this research study?
Well, pharmacologically, obviously, we are, I would say, confirming a lot of the proteins that we're identifying through our proteomic studies, through our protein-protein interaction studies, as well as through our global phosphorylation studies that have identified different kinases. And that's very exciting, but to really nail down, are these specific proteins involved in infection, you got to do the genetics and then do the chemogenetics, obviously, right? So this is fantastic, and that you guys are helping us now to delete each one of these genes in an incredibly a fast way using CRISPR in A549 cells and CACO2 cells and other cells we're exploring. So the goal is to do single knockouts and then double knockouts to look at functional connections between the genes, adding to drugs doing the chemoepistasis analysis to really confirm that the drugs are targeting the proteins which we think are happening. And again, getting these cells, I know you guys are shipping these cells around the world, to our collaborators, specifically in Paris and New York, where they have the virological assay set up, and they're starting to test which knockouts actually are in fact impinging on infection. And then we can marry that data with the pharmacological information that we're collecting as well. You know, normally I would think that it was interesting, you generate a protein-protein interaction map, you do the genetics, you do the structure, then you go to the drugs. But I mean, I realized in this case, and we're going to start doing this across the board as we generate these maps, not just across different viruses, but other disease areas. We're in parallel, we'll do the protein-protein interaction maps, and then the genetics, and then the pharmacology as well, and the structural biology. We'll push on all fronts from day one and put that information together. So this is what I've learned over the last few months. And to me, that shows we could go even faster as we start to tackle other diseases in the future.
Great. So Jacqueline, I actually had a question based on this. Do you think this is a blueprint for how cross-institutional academic industry collaborations can work together in agile fashion in the future?
Yeah, I think not only a blueprint, but our hope, right? Because as we've seen how fast this has developed, the question keeps coming up, well, why can't we do this for other diseases, right? Whether it's cancer or some other virus or whatever kind of disease state. And so it would seem crazy not to adopt something so successful moving forward, right? I think human altruism, as someone just said in the chat, kicks in at this time, and everybody wants to participate in something like that. But I think we need to tap into that, understand what works, and sort of formulate it in a sense. That said, I think that the scientific community is probably the best to do that, because everywhere I've been in the world, there's such an openness to ideas, discussion, and coming together for solutions. So I think this is absolutely the blueprint going forward.
That's good to hear. We actually, few folks, Molly, Alan, as well as Travis, who actually is our head of research, both are asking the same question. So it's good to see that people want to move forward in this direction, as this really does help in terms of agile fashion and how fast things were brought to light.
I guess I'd just like to ask too, Nevan, if these ongoing studies are successful, how do you feel like it's going to impact the current pandemic crisis?
Well, I mean, at the very least, we've generated this data. We're generating more data. We're getting it out to the scientific community. We're getting reagents out to the scientific community as well. So in that regard, we're helping expedite research around the world on COVID-19. And the more we know about this virus, obviously, the more likely it is we are to develop treatments. So I'm excited about that and continuing our work there. But then just some specifics. There was a piece that came out in Nature Biotech a couple of weeks ago where they alluded to our map, and they actually said there has been 13 clinical trials that have already started based on connections from our map. And to me, that's very exciting. And working closely with scientists here like Brian Shoykat and Kevin Shoykat, we're going to be starting a number of clinical trials ourselves in collaboration with a number of the companies that Jacqueline talked about. So here's hoping that one of these provides some success, if not one of these other clinical trials that are ongoing around the world. And personally, for me, I think what's going to happen, just like what was successful with HIV, I think we need a cocktail of drugs here. One may not do it. You may have to do 2 or 3, and maybe one against the virus, one against the host. So maybe one of ours in combination with remdesivir, which targets the virus itself. I mean, there's some positive results come out from remdesivir, but it's definitely far from a magic bullet. So maybe combinatorial studies will be needed in order to come up with a cocktail that will provide some relief as the world, or the scientific world, tries to come up with a vaccine, which I think is going to take a little longer.
Fantastic.
So we got a question from the audience. Go ahead, Jared.
Yeah, so of course, we had a question from the audience. Remember, you guys can submit these. If you're coming to office hours, you can submit these beforehand, and we'll incorporate them into our slides. But can these methods be applied to understand and develop therapies for other emerging viral infections?
Absolutely. I mean, I showed you that map earlier where we've used these approaches across many different viruses. And as I said before, our approach has been generate the map, do the genetics, do the structure, get the mechanism, and then go to the drug discovery. But the fact that we could move so quickly here, going from the map right to drug discovery, to me means we should have been doing that all along, and we're definitely going to be doing that going forward. And the other thing I'd say on this is that each data set from each virus informs the next. Similar sets of human proteins that are being targeted by HIV, by coronavirus, by Ebola, by polio. And the problem is the scientific world doesn't see that, back to this point of being siloed, because people don't talk, when you can bring people together and use technology that's disease agnostic, you start to see these connections across these different viruses, and not just across the viruses, but across different disease areas. You see the same genes being mutated in cancer that are hijacked by coronavirus. And that's why we're looking at all these anti-cancer drugs, because the biology is the same. So as you make these connections around the world, you bring not just people together, but you bring, the knowledge across these different disease areas, so that we could be more effective attacking all diseases in the future.
Yeah, I think that's a really great point about the worldwide. I mean, you can see it from this question that I forgot to mention was from University of Santo Tomas in the Philippines. So it really brings that whole, reaching a diverse audience and making sure that everybody's on the same page. Yeah, and if you guys are interested, you can always check out what Synthego is doing at synthego.com/coronavirus, and you can see our latest updates on our progress.
All right, so I know Jacqueline and Nevan, you guys are extremely busy, and maybe you have to run really soon. So before we wrap up here at CRISPR Office Hours, we just want to thank you graciously once again for coming on and telling us about the work that you're doing. It's really fantastic, transformative, and I think it's really going to help the world. And we're really excited to have been collaborating with you so far. So thank you.
Thank you so much. We're extremely enthusiastic to be working with you, and I think we both loved participating in this. This was fun.
Yeah, thanks for the opportunity. As Jacqueline said, we're super excited to continue our collaborations with you.
All right, fantastic. So I know you guys might have to run, so feel free to drop off if you need to, and we'll look forward to talking to you soon. We just wanted to remind our audience that we've been working through this model of how research may pan out during COVID-19, and as we see the economy start to open up now, we're seeing a transition of people gradually entering back into the lab. And I think we all just need to realize that there's a possibility we could see a second wave, and really the development of therapies is just as important as ever. So we'd like to, again, thank our guests who are really making some significant scientific contributions during this time. So maybe, Aditya, can we move on to see who's joining us next week?
Yeah.
All right, so next week we're actually going to be meeting some Synthego people. So we'll be joined by Jake Johnson, who is the VP of Operations at Synthego, and also Antonia Chair, who is our Senior Director of the Engineered Cells Facility at Synthego. And so they're actually going to be telling us about how a biotechnology company, how a genome engineering company like Synthego can actually stay operational during a worldwide pandemic. So it'd be really interesting to hear about how they've had to adjust the way that they do things, and how Synthego as a company, and maybe as a model for other companies, has been able to shift some of their resources and also understand how to work with people externally during this time.
And then real quick, I want to let people know you can get your Keith, Com, and CRISPR on shirt at synthego.com/com, and we'll send it to you guys. And as we're wrapping up today, we actually want to take a minute of silence to recognize and support the events that are happening across the country to end systemic racism in our community. So just want to take a minute of silence to do that here. All right, thank you for that. And again, we want to say thank you to our panelists, our guests, Nevan and Jacqueline, for joining us, and excited to have CRISPR Office Hours next week. And I hope everyone has a good rest of the week and weekend.